Good. Hello, everyone. Good afternoon. I'm Wenjing Ma from University of Washington from Rob Sinovik's group. I'm currently a fourth-year graduate student, so I'm glad to be here to present one of my recent projects on the tile-based analysis of GC GC-TOFMS mass spec data of SPME-simple volatiles produced from the Pseudomonas aeruginosa at Aspergillus fumigatus. So this project is collaborated with Ting and Jimison from the Burge and Rajakovic groups, also from UW. A really brief introduction. So we're particularly interested in these two species because they are major pathogens found in the lungs of patients with the cystic fibrosis, and their coexistence is associated with worse lung functions and poor clinical outcomes. So therefore, this project is mainly focused on studying their metabolic interactions. Let me introduce our experimental methodology. So to begin with, good, I got it back up. Okay, to begin with, we have four simple classes, and we have four simple classes. Those devices are the SPME device prepared using the microfluidic device, and we're gonna store them in the SPME vials, and their headspace will later sample for analysis. The first class we have is the medium control as our nutrient source, and the second class is the only Pseudomonas aeruginosa. I will call it PA for short. And the third class would be the Aspergillus fumigatus, I would call it AF. And the fourth class would be their co-culture. So for this project, our primary goal is to discover analytes that are statistically different across all four sample classes using the comprehensive gas chromatography with time-of-flight mass spectrometry and followed by the chemometric analysis. In our work, we use the Paxos BT system, and this time-of-flight mass spectrometer can achieve 500 spectra per second collection. It provides a third dimension of data, the full mass spectrum, then we can use for analyte identification. So it's pretty powerful. However, it also introduces really complex data. So for a single chromatogram, including the separation dimension in both first dimension, second dimension, plus the TOF mass spectrum, a single chromatogram could contain up to 10 to the 6 data points. So, and this data even increases with multiple samples, leading to a 4-dimensional data set. It's really hard for people to interpret manually. So that's why we need apply the data reduction strategies. So first of all, we can try create individual peak tables, but this might cause the alignment issue due to the retention time shifting across chromatograms. Or we could try average our chromatograms, but this may also lead to our peak broadening, which further reduces our peak resolution. So we end with using a discovery-based feature selection method, which is the tile-based feature ratio analysis. It uses the F ratio. It's calculated as between class variance divided by within class variance. I will explain this concept using one-dimensional data first. So here would be our one-dimensional chromatogram for those two analyte peaks. They are shown in both classes. And for the analytes in the middle, those three analyte peaks only shown in class A. So when we calculate the F ratio for every data point along the first dimension, so for peaks with high F ratios, they are actually correspond to those analyte peaks that differ the most between two simple classes. So overall, the F ratio analysis will generate a ranked hit list that including all the analytes that are statistically different in concentrations between simple classes. And then now we move on to our two-dimensional data. The chromatographic signal is actually dissected into small rectangular sections. For each one, we call it a tile. And for this, we apply a 4-grid scheme. Grid 1, 2, 3, 4, they are shifted by half a tile size in order to discover all the analytes optimally. And also the F ratio is calculated for each tile on a per mass channel basis. So finally, we're going to end the hit list, including rank by the F ratio, and also we got the hit pin location. We could do further identification and quantification. And this method was further acquired by the Lico company and implemented in the ChromaTOF Tile. And after we processing ChromaTOF Tile, we're going to have a hit table, including all the possible hits. And for specific hit, we can check or look at a selective signal at specific mass channel that pass the user-defined F ratio threshold. And after removing the artifacts, the column bleed, the redundant hits, we can focus on those hits for further identification and quantification. So in ChromaTOF Tile, it also can generate PCA squares plot, providing a visual confirmation for the class separation. Okay, so now let's look at our raw GC-GC chromatograms. So here I show one representative replicate from each of 4 simple classes. As we expected, the media class, the media control, has the least signal overall compared to the other 3 simple classes. And now, as we can see, it's really hard to tell the differences across the other 3 classes based on the raw chromatogram. So that's why we need to apply our tile-based Fisher ratio analysis. So for this analysis, we discovered a total of 576 hits. I will show 2 example hits as example. So for figure on the left, we call it ruler plot. They are generated by summing the tile signal along the first dimension. And for the bar chart showing on the right, they are showing the sum tile signal across simple classes and also across 6 replicates. As we can see, their signals are quite consistent across replicates. We would say, oh, this design gives us a good reproducibility, not only for our device fabrication, also our data collection. And for specifically for this hit number 1, we can see it has high signal in our media class and showing lower signal in the other 3. So this analyte is more likely to be an analyte that consumed by both PA and AF culture. In contrast, for hit number 3, it has high signal in PA and co-culture, so we can guess, oh, this analyte might be produced by the PA culture. As I mentioned before, the tile-based Fisher ratio analysis can discover all the analytes that are statistically different across simple classes, but it's hard to tell which hit is higher in which 2 simple classes like PA and co-culture. So that's why we follow up a pairwise t-test. So this will allow us to find out which classes are actually differ. So for example, we have 4 total simple classes. We're going to end with 6 pairwise class comparisons. And after reducing 6 pair t-tests, this would be the significant result we got. For example, in the comparison between PA and their co-culture, we can see most analytes with higher signal in the PA monoculture. That means PA, those analytes are dominating And now let me switch gears. After we implementing the tile-based Fisher ratio analysis coupled with the pairwise t-test, let's explore more appropriate experimental design approaches for generating high-quality, high-throughput data generation. So ideally, we would have multiple biological replicates, but in reality, that's not always be the case because we're constrained with simple availability, the cost, and time. So under our practical constraints, we have limited samples, and we have a high reproducibility requirement, and also sometimes we have high precision demand for our devices fabrication. Or for this SPME data set, we have a time-sensitive requirement too. So we need to choose the correct, really right experimental design approach in order to collect high-quality data more efficiently. So when we do not have replicates available, we actually can perform a tile-based coefficient of variation analysis. So this will utilize a metric called coefficient of variation, CV in short, calculate standard deviation over the mean value. So this metric will discover analytes that are statistically different concentration changes with only one replicate needed. So to demonstrate, we perform our this analysis using one replicate from each of 4 sample classes. We apply the ChromaTOF Tile using the coefficient of variation method to obtain a single comprehensive list, including all the analytes with up and down regulations from each of 4 sample classes. So now I will show several example heads. First, those are two example heads with the highest signal in our media class. For those match values, they're not always about 800. So for further study, we might apply a chemometric tool to enhance their analyte identification. But let's look at their signal. So for example, those two hits actually have high signal in the media class, so we can also infer those two analytes might consumed by both PA and media cultures. And here are another 2 example hits with high signal in our PA monoculture. Specifically, this is only shown in PA monoculture, so it might produce by PA and also not showing it up in the co-culture, so this analyte might be constrained by the presence of the AF culture. And for the other analyte, we can see a show up in both media and PA monoculture. Then we can see, it's not being affected by the presence of the PA culture, but it's constrained by our AF culture. And here are another 2 example heads with high signal in our AF monoculture. And this is actually a 1 octane 3 node with a quite high match value, about 800. So we reference, we confirm this analyte is actually a known volatile produced by the AF monoculture. So it's kind of a validation check for our data analysis and identification. And here will be the last 2 example heads with high signals in the co-culture. Last but not least, in addition to the facial ratio analysis and also the coefficient of variation analysis, in ChromaTOF Tile, we can also implement the tile-based pairwise analysis using the metric called fold change. For example, we can perform this analysis between only media source and media control group and our monoculture to discover which analytes are being generated by the PA or consumed by the PA. And we also got the hit list similar before, but this one will be ranked by the fold changes. So in summary of our pilot study, first of all, if you have well-known replicates, the tile-based facial ratio analysis would be a good choice. However, if you, under reality, you do not have replicates, you want a high throughput experimentation, then the metric coefficient of variation would be a more suitable choice. So all in all, the choice of the metric selection, amount F ratio, coefficient of variation, or fold change, it actually depends on the experimental design you want to achieve. And for further studies, we're going to apply chemometric tools to improve our analyte identifications and bring into a more comprehensive analysis across different approaches. Yeah, that'll be all. Thanks for listening. Any questions?